Okay, today we are going to be taking care of diffusion, cellular diffusion. We're going to be finishing up the cell in a few days. But in order to understand tissues, which is what we will be getting into later this week, you have to understand a little bit of diffusion, which is how particles move across the membrane from one area to another. Remember that a membrane is no more than a barrier separating two areas, and it can sometimes let things through. The wall behind me is what distinguishes my room from the hallway. The wall to your left is what distinguishes my classroom from my closet. Now, when it comes to these membranes, some of these membranes can be semi-permeable. A semi-permeable membrane, it has that look, and we know semi means half. Permeable means things can pass through. So typically a semi-permeable membrane allows certain substances to go through, disallows others. So if you take a look, I'm going to pause this in a little bit so we can work on your study guide here. Let's talk about a plasma membrane for an example. Let's see what you remember from a few days ago. Is the plasma membrane polar or nonpolar? It is nonpolar. Very good. So what types of substances would you expect to have the ability to be able to move across a plasma membrane from one side to another? Polar substances or nonpolar substances? Only nonpolar substances will be able to pass through. Okay. So that's the main gist of diffusion. There's two different types of diffusion. There is passive transport and there is active transport. If you want to make a T-chart to organize your notes, you're welcome to. I will. Okay, as I mentioned in class before, I own a paddleboard. I go around Davis Island, Harbor Island, downtown, Riverwalk, all those places. And if the current is with me, I don't have to paddle. I just can stand there. I can sit there and just take in the scenery. Passive transport requires no energy. And Scout, what is that cellular energy known as? ATP. You do not need adenosine triphosphate. It's passive. In passive transport, particles diffuse from a high concentration to a low concentration. We're all going to go to Chris's house tonight, and he's going to cook us some chicken nuggets. He has enough for everybody. So we're all going to hang out in his living room, kind of talk, do our homework together while he's cooking us dinner. His kitchen is like a solid 30 feet from where we are going to be. and he gets started cooking all by himself. And then a little while later, we all start to smell that aroma of chicken nuggets and food. The kitchen had a high concentration of that aroma, that smell. The living room, the hallway, the laundry room, upstairs, the bedrooms, the bathrooms, they all were low concentration. The house is slowly starting to fill up with the smell of food. That is passive transport. That is passive diffusion. It doesn't require energy, and it goes from an area of high concentration, like the kitchen, where the food is, to areas of low concentration, where the food isn't. Living room, bedrooms, laundry room, hallways, bathrooms, so on. So I know that if you're online, you can't see this. I'm sorry about that. But if I have a beaker of water right here, guys, let me empty this out and start over. Don't worry, it's on my refrigerator. How would you describe the concentration of food coloring in this beaker as it stands right now? Low. It's pretty much absent. How would you describe the concentration of food coloring in this little dropper? High. Very good. So now, how would you describe the concentration of the top of the water? How about the bottom? Low. Good. What's going to happen? It's going to go from high to low. When will it stop? Anna? When you have achieved equilibrium. That's when diffusion stops. That's when the smell of chicken nuggets will stop filling out Chris's house. When the whole house smells like nuggets. Until equilibrium is achieved. This not only works with food coloring and smell of chicken, it works with sound. It works with light. Yes, it works with smell. It works with a lot of stuff. Anything that can travel across the membrane from one region to another. So because we're going from high to low, we say that we go down the concentration gradient. So here's what we just did. Here's a beaker, and I dropped in some food coloring right there. That area right there is going to be really high. Give it a couple minutes. And before you know it, the whole thing is going to be equalized. That's diffusion. This right here, that little drop is the high concentration. All of this area is low concentration. It is going to diffuse from high to low until the entire area is equal. That's diffusion. More specifically, that's passive diffusion or passive transport. There are three examples. One, simple diffusion. This is the one that students tend to forget the most. Give yourself a little extra. There's no fancy name to it. There's the osmosis. That's a tricky name. I got to think about it. Facilitated diffusion. There's no one. This is simple diffusion. This is one of the three. This is where small particles diffuse across the membrane. But I'm going to make an asterisk here, guys, and I'm not going to write this down. I'm going to just tell you because this has to go without saying. The polarity of the particle must be the same as the polarity of the membrane. Like you said earlier, nonpolar particle can diffuse across a nonpolar membrane. A polar molecule can diffuse across a polar membrane, but you can't have opposites. You cannot expect a polar particle to diffuse across a nonpolar membrane or vice versa. It's not going to work. Number two, facilitated diffusion. To facilitate means to help. If you have a learner's permit and your parents are teaching you how to drive, they're facilitating your driving ability. They're helping you. Facilitated diffusion is where large particles use a protein channel to diffuse across the membrane. Some particles are just too big to just slip through the membrane. So they need a tunnel. They need a channel protein. And the third one, which we need to spend some time on, osmosis. What is this the diffusion of, Eva? Don't whisper to her, Isabella. What comes out of those drains next to, or those faucets next to you? Water. It's the diffusion of water. And this should also go without saying, because this is passive transport, it's high to low. These are the three examples of passive transport. If you're in AP Bio with me, then you've already heard this on Friday. We just so happen to be learning the same thing that's going to be stopping this week. No, because in AP Bio, we're going to be getting into the mitochondria and the chloroplast after your test next week. And in this class, we're starting tissues. And that's where it's going to diverge forever for the rest of the year. So this is the last week where AP Bio and anatomy are going to be aligned. All right, everyone. Now, I'm going to present this information to you for osmosis in an easy way, and then a more critical thinking way, okay? So, Jack, you're awfully quiet over there. Let's see if you can help me figure this out. This is a membrane. Jack, this membrane has aquaporins. Now, I've not told you all this yet. An aquaporin is a protein channel that allows water to go through. Okay, water can go through it. We have 97% H2O here, and we have 3% sodium chloride. Sodium chloride is table salt. And over on the right side, we have 90% H2O, and we have 10% sodium chloride. Jack, which region has more water, left or right? Or let me rephrase that. Which of these has a higher concentration of water? The left. So which way is water going to diffuse? Left to right or right to left? Left to right is the easy answer, and that is correct. So Jack's not a rocket scientist, but he knows that 97 is more than 90. Simple transport is from high to low. It's going to go from left to right. That's the easy way. Now let's change it up a little bit. There's our membrane. All right, here we go. 0.2 molar, 0.8 molar. Now, if you're a little rusty on the chemistry lingo, let me jog your memory. Molarity is the measurement of concentration of solute in a solution. A solute is the thing that gets dissolved in a solution. If you were in the Gulf of Mexico this weekend because you went to the beach, the salt in the Gulf of Mexico is the solute. The salt water is the solution. The water itself is the solvent. So in this particular example, I'm going to tell you that sodium chloride, table salt, is your solute. Jack, which area is more concentrated in salt? The left or the right? The right. 0.8 is more than 0.2. So Jack, this is where we got to get into our line of thought here. Jack, if there's more salt concentration on the right, what is there less of? No. What else is in there besides salt? There's less water. Okay. So here's how I think of this. If this has more solute, or in this case is salt, there's less water. Now you always have to have a basis of comparison, like more salt and less water than what, Mr. Thorson? The left side. This side has less salt, so it has more water. So Scout, which way is water going to diffuse? From left to right or right to left? Left to right. That way. So there's two ways to look at this, guys. Water, you can either say water diffuses towards the region with less water, or water will always diffuse towards the region with more solute. I've said this earlier, you are not rocket scientists, but I think you can determine that 0.8 is greater than 0.2. Water will always diffuse towards the region with a greater molarity every single time. It will diffuse towards the area with a greater concentration of solute. So let's take a look at this. Oh, I don't want to get rid of that. There we go. Okay, if you were in AP Bio on Friday, you remember this. Here's what I want to ask you guys. We're not going to do hyper, hypo, or isotonic yet. I want to ask you about this page right here. Let's make sure you're sure. Okay. Let's just call these beakers 1, 2, 3, 4, 5. I just want to ask you a simple question. I don't want to ask you about hyper, hypo, or isotonic yet. We haven't done that yet. We'll get to it. Those little yellow baggies, pretend those are cells. And the solution that they're sitting in is the solution. Just say solution. So let's take a look at beaker number 1. The cell is 0.2 molar. The solution is 0.6 molar. Chris, what has a greater molarity? The cell or the solution? The solution. That's easy. 0.6 is greater than 0.2. And if you remember what I just said, water will always, no exceptions, always go towards the region with a greater concentration. Melanie, paying attention? I've been doing that since class started. I don't know what you're doing. Chris, which way is water going to flow in cell, or excuse me, in example number 1? Into the cell or out of the cell? Out of the cell. Eva, how about number 2? 0.2 versus 1.0. Which one has a greater concentration, the solution or the cell? So which way will water flow? Into the cell or out of the cell? Good. Let's see. Sam, beaker number 3. 0.2, 0.2. What's the flow? It's equal. Good. We'll get to that momentarily. Alyssa, number 4. 1.0 molar inside the cell, 0.2 molar outside the cell. What has a greater concentration, the cell or the solution? And so which way will water flow? Into the cell or out of the cell? Correct. Last one. Let's do Sloan. We have 0.2 versus 0.4. What has a greater concentration?
The solution.
So which way will water flow?
To the solution.
Correct. All right, guys, you're 5 for 5. We're doing well so far. So let's finish off osmosis with hypotonic, hypertonic, and isotonic. If my blood sugar levels were considered hyperglycemic, is that above normal, normal, or below normal? So what does hyper mean? Hyper means above. If a cell is in a solution that is hypertonic, I want to reiterate that. It's the solution that's hypertonic, not the cell. A solution has more, wrong word, has greater solute molarity than a cell. So here's my beaker. Here's the solution. The solution I'm going to say is 0.8 molar. Somebody tell me a molarity for the cell that would make this solution hypertonic. Any number. You got plenty to choose from. Just tell me one that would make sense. Anybody? 0.2. Fine. So will water exit the cell or enter the cell? Or will there be no change? It'll exit. Because the molarity of the solution is greater than the cell, water is going to exit the cell. The cell will shrivel. This is called crenation. This is why you don't drink salt water when you're stranded on a boat or an inner tube in the middle of the ocean. You die of dehydration. It seems weird. How do I die of dehydration from drinking water? Here's how. If you put a bunch of salt on your tongue, what happens to your tongue? It gets all dry. Because what do you think has more sodium, more salt? Your tongue, in the cells in your tongue, or this big pile of salt you just put on your tongue? Big pile of salt. So water will always go towards the region with a greater concentration of salt. If you covered a slug in salt, what happens to the slug? It dies. How? How does it die? Die of what? It dries out. All that slime and moisture in a slug is going to basically, for lack of a better word, get sucked out of it because that salt is absorbing all of it. So whenever you drink salt water, the water that's going to be in your stomach has more salt than your surrounding tissues and organs and blood. And so all the salt in your tissues and organs and blood, because you do have salt in your body, is going to leave those areas and go towards your stomach. So it's basically dehydrating your tissues. That's how you die of it. All right. Let's go to hypotonic. What does hypo mean? If my blood pressure was hypotensive, is that below or above regular pressure? Below. So hypo means below. Never forget that. You've seen that term a lot this year. In a hypotonic solution, the molarity of solutes in a solution, SOLN is how a lot of science nerds abbreviate solution. Any solution is less than the molarity of the cell. Okay, so here's my beaker. Here's some water. Here's my cell. My solution is 0.8 molar. Somebody tell me a number that would make this statement true for this to be a hypotonic solution. 3? Not 0.3, but 3.0? Yeah. What? 3.0. Okay, good. 3 molar. Yeah, that would work. That's a huge difference. So which way is water going to flow? Into the cell or out? Into. What could that cause the cell to do? Yeah, big time. I mean, that 0.8 to 3, and that's a huge difference. When will the diffusion stop? When it's equal. Yeah, when it's equal. 0.8 is going to go up and 3 is going to go down until it's diluted. The cell will swell. Sometimes it might burst. This is called lysis. It might split. I had a student say a long time ago, hypo like a hippo, swells up like a hippo. And then we have isotonic. I think you might know what this is. Have you ever done a wall sit before? That's an isometric workout. Some people, when they work out, they're like this. 1, 2, but look at me. Relaxed, contracted, relaxed, contracted. Your body, your muscles get a break, like a fraction of a second break. When you're doing isometrics, like a wall sit, it's full contraction, no rest. So you're not changing though. You're not moving your arm. You're not moving your legs. You're unchanging. Iso means unchanging or the same. So this is where the molarity of a solution is equal to the molarity of a cell. So here's my beaker. Here's my cell. Solution is 0.8 molar. Somebody tell me, what would the molarity of the cell be in order for this to be isotonic? It would have to be 0.8. And there would be no net movement. They're equal. So water would go in, but the same amount of water going in would have to equal the water going out. So if I have one arrow going in, I have one arrow going out. One arrow going in, one arrow going out. It's equal. No net change. I have 20 students in this class. If 20 students exit and 20 students enter, I get no net change. Okay. We're going to take a break from the notes. I'm going to hit pause. And I want you to take a look at your packet. Please look on number 38, or excuse me, page 38, number 8. Everybody there? Here we go. Figure 3-4 shows a semi-permeable sac containing 4% sodium chloride, 9% glucose, and 10% albumin. You don't have to worry about what albumin is. That's not the point. The point is that you have a solution with salt, glucose, and albumin. It is suspended in a solution with the following composition: 10% sodium chloride, 10% glucose, 40% albumin. Assume the sac is permeable to all substances except albumin. This means albumin cannot cross the little membrane that makes up that sac. Using the key choices, insert the letter indicating the correct event in the answer blanks. Okay, guys, let's look at glucose. What has a greater concentration of glucose, the solution or the cell? So we're going from high to low here. So which way is glucose going to diffuse? Into the sac, out of the sac, or does not move? It moves into the sac. That is correct. Glucose is 10% in the solution, 9% in the cell. We're going from high to low. Let's go to number 2. Water. Okay, let's figure this out. But Mr. Dorsey, water's not listed. We can figure it out. If the solution is 10, 10, and 40, that's 60% of the solutes. That means that the solution is 40% water. And in the sac, we have 10, 9, and 4. That's 23% solute. So that should be 77% water. So the sac is 77% water, and the solution is 40% water. Which way is water going to diffuse? Out. 77% water inside, 40% water outside. You're going to go from 77 to 40. It's moving out. Albumin. What's that going to do? Now I see that's 40% outside and 10% inside, but what did the reading stipulate? Albumin can't go through the membrane. So let me phrase it this way. Let's just say albumin could go through. Let's just say, don't write anything down. Which way would albumin go? Towards the solution, which is out of the sac, or into the sac? It would go in, but it can't move because the reading says it can't go through. It's not permeable to albumin. So you would say it does not move. And then finally, we have sodium chloride. We have 10% in the solution. We have 4% in the sac. Which way is it going? Into. Nice. Okay, for number 9, you need to look on page 39 as well. So open up your page, unfold it so you can look at both images. I want you guys to take a look on page 39, figure 3-5. Now I want you to tell me in circle A, you're looking at a microscope and you see those cells. They look pretty shriveled to me. What type of solution must those cells be sitting in? A hypertonic, hypotonic, or isotonic solution? They're sitting in a hypertonic, which means the molarity outside the cells is greater than inside. How about cell B? Looks like there's no net change. Iso. And how about cell C? They look pretty swollen. Hypo. Okay, good. Let's answer number 1. Which microscopic field contains a hypertonic solution? Survey says A. Good. The cells in this field are said to be, what's that C word that I told you? Crenated. Yeah, crenated. I told you crenation is where they shrivel. They were crenated. Number 2, Alyssa, which microscopic field contains an isotonic bathing solution? Correct. And isotonic you should have in your notes. I'll let you fill that out on your own. Don't just say equal. Say what is equal to what. Make sure you say it like that. Don't you equal. I mean, that's middle school. What is equal to what? Well, That's the consequence, but iso has a specific meaning. Number 3, which microscopic field contains a hypotonic solution? I think we can narrow it down to C. What is happening to the cells in this field and why? What is happening to the cells? Yeah. They're going through lysis. Why? Why? Good, good. So eventually we get to the point. So she says the cells are swollen. They're going through lysis. I said, why? She said, because water is rushing into the cells. I said, why? She said, because the cells have a greater molarity than the solution. Eventually the answer can be revealed. Good job. All right. I'm going to hit record again, guys. We have just a itty bitty to do with active transport. It's actually very quick, not nearly as long as passive transport. So here we go. Okay, now that we are finished with passive transport, let's go back up and fill in active transport. In active transport, energy is required. If I were to go paddleboarding and I had to paddle against the wind and the current, I'm in for a workout. Because I'm going against the wind, I'm going against the current. If I could be swept out to the bay if I don't paddle hard enough. I got to go against, I'm going to need that energy. In this form of transport, particles diffuse from a low concentration to a high concentration, so it's reversed. Oh, sorry, cruise control there. From low concentration to high concentration. So the best way I could describe this is, imagine if I could take all the food coloring out of that water and put it back into the dropper. That would be going from a low concentration to a high concentration. You're making a high concentration even higher. So we're going up the concentration gradient. The examples that are mentioned in this topic is the sodium. Sodium is Na, potassium is K, sodium potassium pump. This is found in your nervous system. It helps transfer electrolytes into and out of your neurons. We have endocytosis. What do you think this is? Stuff coming into the cell or stuff exiting the cell? This is bulk transport entering the cell. What do you think the opposite of endocytosis is? Yeah, exocytosis. Exocytosis is bulk transport exiting the cell. We have phagocytosis, which is what your white blood cells do whenever they see a bacteria. It is the movement of solids. And pinocytosis is the movement of liquids. Okay. All right, so we're done with your notes.